Hello everyone. Today we will be discussing about PerkinElmer's multi-mode plate reader portfolio. PerkinElmer was the first company to come up with the concept of multi-mode with the Victor system that was introduced in the early 1990s. Going on with that strong history of multi-mode detection, PerkinElmer today can offer different kinds of system like the Victor Nevo, which is the basic model which can be used in every academic research institute for multiple user base. So now I will introduce the Ensight system to you. Ensight is a wonderfully prepared system which gives you a combination of quadruple monochromators which can be used for absorbance, fluorescence and for time resolved fluorescence technology, and it also has a dedicated close proximity PMT or photomultiplier tube that can be used for luminescence and alpha technology. This machine becomes a brilliant bridge between the multi-mode detection systems as well as the high content screening system, because it also comes with imaging with an SCMOS camera and the LED unit that can be easily done using this particular instrument. Now I will be demonstrating PerkinElmer's orthogonal approach with this particular plate, where we have three different dyes like blue, which is DAPI, then we have Alexa 488, which is green, and finally we have Alexa 47, which is red, and we have stained the HeLa cells with these particular dyes so that we can observe two different concepts in one single experiment. We have loaded the plate into the instrument. We will start a new experiment. We will give this a particular name, like we can say demonstration of fluorescence. The plate type will be selected as Cell Carrier Ultra, as this is the plate that we have used. We can select the measurement sequence as per our requirement, which will be fluorescence intensity. First of all, this is cell based experiment, so we will select the bottom mode of imaging, and the excitation wavelength for the green channel is 488 and emission will be 520. We have selected both of them. For the next step, we can select the imaging mode that we require. Once we have selected imaging, we can select the channels that we require. We can select the blue channel, we can select the green channel, and finally we can select the red channel. Once all our channels have been selected, we can select the analysis method that we need. So let us go into the imaging analysis which we are going to require. We will require object counting and we will need object intensity analysis. We have selected both of them. Let us configure the settings. The object detection will be using the DAPI dye, so it will be blue. For our results, let us take the number of objects or the object count. We can have the intensity in all the three channels also available for us. We have selected all these parameters. Let us do the final step and select the wells that we are going to now read. Our samples will be located from C4 well to C12. We have selected those samples. We can save this particular experiment. Now that we have set all our parameters, we can go to run protocols and we can simply start this particular experiment. Let's click on start. As we can see over here, we have got the images in three channels. This is blue, then we have the green channel, and finally we have in the red channel. So we have got images in all three modes. Now we will do additional analysis for this. So let us start the analysis process. Let us add an analysis sequence. We can select the channel, we can select the results of our interest, and we can tell the instrument to calculate these results. And as you can see, we have got the object count over here. This is the area of all the objects that we have got, and finally this is the intensity of the blue channel, the green channel, and finally the red channel within this particular wells.